there's been a lot of debate in the scientific community about geronto drugs, molecules that can delay aging and send the biological clock back. So, of course, if you don't have a reliable clock that measure aging, we can't do anything with these molecules. So it's useless to develop drugs for aging if we cannot measure aging. And years ago, I think a good maybe 12, 15 years ago, Steve started to work on what is today the most reliable, the most trustable biological clock that we have, which is based on epigenetic mechanism that we'll explain about. So Steve Horvath is really the founder of the new science of molecular clocks of aging. And today he will speak us, he will give us his last result. He's from Altos Lab, Cambridge. Thanks, Steve.
Yeah, thank you, Alberto, for inviting me. This is a phenomenal conference. I loved all the talks. I will talk about my favorite topic, which is, of course, how to measure time, how to measure mortality risk with methylation. And just to step back, it's all based on the DNA molecule. The DNA molecule carries our genetic information. However, we now know that it also carries information on time. I want to briefly show you a textbook image of the DNA. And you will notice that the DNA is wrapped around proteins. These proteins are known as histones. And when we talk about epigenetics, we refer to histone changes, but also methylation changes. But what do these changes do? They fundamentally determine whether the DNA is condensed together, closed, or whether the DNA is open, whether genes can be active. And so these little balls you see are all about histones. And I briefly want to mention that recently Lucas Paulo de Lima published clocks for histone marks in humans. So for most of the histone marks that were even mentioned today, we have a histone clock. So why is it that we don't really measure histones in population studies? Well, because it's, first of all, difficult to measure, it's more noisy to measure, and it's expensive to measure. However, just as a proof of concept, this is an important study because it shows you can build clocks based on histone marks, pretty much any histone mark. However, I will now come to cytosine methylation, mainly for historical reasons. It can be, and technical reasons. You can measure it relatively cheaply and robustly. And one insight that came from methylation studies is that you can really measure time from cradle to grave. You can measure time in an embryo, and you can measure time in middle age and centenarians. And earlier today we heard a wonderful talk on Clotho and the mythology around that. But to me, this continuous readout that connects development to aging, this thread of time, is really DNA methylation. Now, I want to show some illustrations. I'll start with this remarkable observation that Italy has many centenarians, and nobody doubts it. Now, you may have heard, of course, of these so-called blue zones. There are 5 blue zones across the world where there are records of exceptional longevity. People live 5 or 7 years longer than the average population. So these are the blue zones. However, the concept of blue zones has recently been attacked by a very thorough researcher. His name is Saul Newman, and he built a strong case that these blue zones may reflect poor reporting or, at worst, pension fraud. In other words, if somebody dies, don't report them, continue to collect their check, you know, or lie about your age. And in the past, we were not able to verify claims of exceptional longevity. However, I partnered with an Italian team, notably Paolo Garagnani, who spoke earlier today, and Claudio Francesco, and many others, to build an epigenetic clock for validating age claims of centenarians. So, in other words, this clock is accurate enough to find discrepancies of 10 to 15 years, you know. There's no question about it. But one thing that came out of this centenarian clock is it was built with cytosines that also relate to aging in children, again highlighting the connection, the universality of methylation age from development to centenarians. Now I want to talk about mammalian species. So as everyone knows, animals age differently. Dogs can live up to age 20. Then conversely, certain whale species can live 211 years. And the remarkable thing is that although we have many different mammalian species that live in different environments, a whale lives in ice-cold water, a bat flies, other species, of course, are exposed to hot or cold temperatures. So very different ecology, and these species are separated by several 100 million years of evolution. But we can build one clock, one regression model based on about 1,000 cytosines, 1,000 locations on the DNA to measure age in all of these species at the same time. So it's not one clock per species, it's one clock that applies to all mammalian species. And we call this clock pan-mammalian clock. And I briefly mentioned that the pan-mammalian clock estimates relative age, which is age divided by the maximum lifespan of the species. And we and others have used this clock to study aging. And one thing that we could confirm, and many of you may know that, but if you connect the circulation of an old mouse to the circulation, the blood circulation of a young mouse, then there is an equalization of age, meaning the young mouse gets older because it's exposed to the blood from the old mouse. It stresses the young mouse. But conversely, the old mouse gets younger. This is known as parabiosis. And I want to use this system briefly to make another point. So here was a collaboration with Vadim Gladyshev at Harvard, and we added a twist to the experiment. So initially, the mice were connected for 3 months, and we then, after we collected blood, and then we separated these mice again for 2 months and let them live their lives. And why did we do this separation? We wanted to see whether the rejuvenation effect persisted or will it go back. And the answer is the rejuvenation effect did not persist, okay? Meaning the old animal that was rejuvenated using this young blood, after separation from its young partner, it aged faster again. And and this is a very general phenomenon actually in the whole aging field. Unfortunately, many interventions work only briefly, you know. It's not like you can do something and then it will go on. More often than not, you need refreshers, you know. Now, conversely, this same has been found with stresses. Interestingly, there are certain stresses that increase your epigenetic age quite quickly. For example, parabiosis increases the age of the mouse, but there are also other stresses, COVID or surgery, major surgery can really increase your epigenetic age in a day or two, to a great surprise to me. But then if you let the person recover, then interestingly, the epigenetic age goes back. So one insight of a fairly recent insight is how dynamic epigenetic age is actually. Yeah. Now, I want to come to another topic that's very popular right now, these ice baths. Many people believe cooling is good for you. And to rigorously study that in an animal system, we looked at an interesting animal known as a marmot, yellow-bellied marmot. And we did find some evidence that during hibernation in the wintertime, when this animal shuts down the metabolism, that then epigenetic aging was reduced in these animals. Now, the effect was weak, and I didn't quite know what to make of it. I needed to see more replication of this effect. I wasn't convinced yet. Now, interestingly, there was a group at Harvard who carried out a very rigorous mouse experiment to study this effect of hibernation, or what is also known as torpor. Torpor means you shut down metabolism and cool down the body temperature. And they developed a mouse model where they stimulated certain neurons in the brain, pre-optic neurons. And when you stimulated these neurons, it led to a lowering of the body temperature of the mouse. The mouse body temperature was reduced. And here, what you see in the scatter plot is how the methylation age of blood in these mice changes across time. And this was a 9-month study where the body temperature of these mice was lowered by this neuron stimulation. And I think you can see that consistently the methylation age was lower, up to 3 months lower, which is a strong effect, you know. And now I show you this picture of this science fiction idea, because whenever you see a science fiction of space travel, they induce torpor in the astronauts to prevent accelerated aging. So maybe there's something to it, you know. Now I want to come to our Grim Age clock. Grim Age is named after the Grim Reaper, which is a medieval figure of death and mortality. And Grim Age is widely used in epidemiologic studies. And it really relates to inflammation and any health behavior that relates to inflammation. So the first speaker today gave a wonderful overview of inflammatory exposures. Grim Age would capture it. I want to add, Grim Age is not an inflammation marker per se. It measures many other dimensions, you know. But anyways, smoking, exercise, vegetable intake, all of this affects Grim Age. By the way, Grim Age is not really a measure of age, meaning your calendar age. It really is a measure of what one calls hazard of mortality, your hazard of death. For the experts I mentioned, it comes from a Cox regression model. Yes. Anyways, so what do we know about interventions that affect epigenetic age? Well, back in 2018, we analyzed the Framingham Heart Study, and we found a very weak correlation between self-reported omega-3 intake and grim age. Weak correlation 0.1. And as you know, these observational studies cannot be trusted. What was really needed was a clinical trial. And 7 years later, I'm very pleased to mention a Swiss professor, Heike Bischoff-Ferrari, accessed blood samples from a randomized controlled clinical trial. This was a flawless study. Blinded. The people took a pill, they didn't know what they were taking. It was a blinded, very well-controlled study of not just omega-3, also vitamin D, and a home exercise program. And the study was noteworthy in regards to another aspect, which are the participants. All of the people who participated were healthy older adults. They were 71 years or older, average age 75, but overall pretty healthy. And the study lasted for 3 years, and sample size 780 samples. And what did we see? So here at the bottom, I show you grim age, the effect, and their different interventions. And for people with a good eye, I mention that omega-3 led to a small reduction in grim age. And the upper panel shows you another widely used mortality predictor, a methylation clock, known as pheno-age. And so both clocks really show that omega-3 supplementation reduced epigenetic aging. I want to say the dosage was 1 gram. I mentioned the dosage because some people have found that if large quantities of omega-3 may lead to atrial fibrillation, so be cautious, dosage matters. Now, the one thing I was quite disappointed in this study is we also, of course, so one disappointment was vitamin D. I take vitamin D every day. There wasn't really a strong effect. The next disappointment was this home exercise program. So these people did some home exercise intervention, and there was no effect. But then one would say, well, maybe they didn't exercise hard enough. More vigorous exercise is needed. And what is the most vigorous exercise you can think of? Participating in the Olympiad, okay? So this is an interesting collaboration with Hungarian professor Zsolt Radak, who had blood samples from a unique cohort. These were now Olympic champions and age-matched controls. And sure enough, the Olympic champions who had won a medal less than 10 years ago, their epigenetic age was lower than expected. So I liked that result. But the interesting finding was people who had gained an Olympic medal more than 10 years ago, they didn't differ from the controls, And that, again, perhaps illustrates this bouncing back effect, You can't just participate in the Olympiad and just assume now you're rejuvenated for the rest of your life, You just need to keep working on it, By the way, I'm getting scary time messages. Should I continue? Oh, okay, yeah. You stop me, okay. Yeah. Now, hormone treatments. Hormone treatments are controversial, but what we have found is that post-menopausal hormone therapy very much slows the epigenetic age of skin cells, more precisely buccal epithelial cells. So these are skin cells. But we never found an effect in blood, disappointingly, And my mother-in-law said, but everyone knows that hormonal therapy keeps your skin young, you know. So anyways, but we find it also with the methylation clock. So you notice a lot of things relate to epigenetic clock. Inflammatory exposures, hormones, temperature perhaps, and so on. And so the question is, what makes epigenetic clocks tick? And we recently published a paper with Madi Mokri, where we listed 5 factors, and I just enumerate them. Cellular replication, the number of mitoses matters. Cellular composition matters. If you analyze blood, the abundance of naive T cells versus memory T cells. And of course, many exposures, smoking exposures, metabolic stress. If you're obese, it accelerates your clocks. And so the clocks differ in their sensitivities. Some clocks really relate to cell differentiation or proliferation, others don't. And that poses challenges for the data analyst. The data analyst needs to very much think about what covariates they put in the model. Here I present a review paper that I published with Andrew Teschendorf on building novel clocks, because the epigenetic clock field is very active. I want to say every other week a new clock comes out, which is wonderful. And here we again reviewed all the factors that are known to influence epigenetic clocks. So now I mentioned circadian rhythm. There's a paper that claims if you measure your methylation age at midnight, you're slightly younger than if you measure it at lunchtime, which is an interesting claim. Yes, and now the other thing I want to mention is that we are now in the era of artificial intelligence, the so-called large language models. We're in the era of deep learning. And of course, most of my clocks were based on so-called penalized regression, which is a phenomenal regression model. But people have used deep learning approaches to build more accurate clocks. So there's a lot that can be done. The biggest distinction between clocks is whether they are meant to measure chronologic age or mortality risk or another readout of biologic age. So you can distinguish them along those lines. I want to mention that we have very accurate predictors of maximum lifespan in mammals. If you give me the DNA of any animal, any mammal, I can give you a rough estimate of its maximum lifespan or its age at sexual maturity or gestation time. So again, methylation encodes these life history traits. And one thing we found with these studies is, again, what some of you may know already, that dwarf mice age more slowly and conversely have a longer maximum lifespan. That's known from the literature. This is 20-year-old findings, but we can confirm it with our methylation clocks. The new frontier in methylation clocks when it comes to different species is to extend methylation clocks to non-mammalian species, ideally all vertebrates. So we have some progress in that arena. We have some prototype clocks for frogs and axolotl amphibians. These clocks are not very accurate, but they show proof of concept. I started earlier talking about female hormones. I want to briefly talk about male hormones, androgens. So there was a wonderful paper by Tim Hoare and Victoria Sugru, who found one cytosine in the DNA that is very responsive to androgen exposure. And it really can measure exposure in multiple species, in mice, in sheep. So for example, here, and they call it the androgen clock. So here, they administered an androgen, DHT, to female mice, and sure enough, the methylation increased in that cytosine. And conversely, you need the androgen receptor, because if you knock out the androgen receptor in these mice, and you then administer the hormone, nothing happens. So this is the one epigenetic clock where we truly understand the mechanism from A to Z. Androgen exposure leads to gain of methylation in this cytosine. I want to end by talking about longitudinal studies. They will absolutely be needed to show causality. In other words, we need to measure whether an increase in Grimm age over multiple blood draws means you have a poor prognosis for mortality. And there was another wonderful Italian cohort, the Interventi cohort. This is a study with Luigi Ferrucci and an Italian team, where they really looked at this question. They had blood samples from almost 700 people, Italian participants, and they had at least 2 blood draws, sometimes 3 or 4, and they could measure rate of change. And sure enough, they found rate of change in these people meant faster decline, earlier death. So in summary, methylation is quite appealing. Why? It's intellectually appealing. The same molecule that carries genetic information encodes time. The beauty of the universality of aging processes. We have one clock that measures aging in all mammalian species. So it cannot just be random noise. There's something really conserved in aging. The next thing that's so beautiful, and that goes to the Clotho story, we can connect the life course. We can connect aging in a fetus to aging in a centenarian. There's something very appealing about that idea. Methylation clocks are ready for human clinical trials. People are using them. They are also used for in vitro studies. Many people apply them to fibroblasts, keratinocytes, any cell culture system. And I'll stop by thanking my many collaborators. Thank you very much.
Very interesting, fantastic. Thanks a lot. So, Annibale, first, poi ci saranno altre domande.
Thank you very much. It was illuminating.
Non è acceso.
It's been illuminating.
Yeah.
My thinking of when you were talking was, what if this methylation profile is more about a stress response more than an aging process? So parabiotic experiment, the fact that the Olympic guys were basically getting back to a normal methylation profile. Is that due to the fact that you are basically looking at a picture of what is going on in that moment more than something that had been changed and then give you some advantage in terms of aging?
Yeah, well, you are definitely right. Somehow these clocks reflect stresses. There's no question about it, you know. See, I think of clocks in certain ways as integrators. Certainly many forms of stress impact on them. But they are also under genetic control and maybe not so much stress related. That's why I kept mentioning embryogenesis, you know, early development, where arguably you don't really have a lot of stressors, you know. So yeah, I don't know. Mm-hmm.
But during stress, you basically switch on genes that are related to stress, like in the fetal genes. Sorry. During the stress, you're going to upregulate fetal genes, like the heart, no?
Yes. No, that's true. Yeah, I agree. Yeah.
Thank you very much.
I agree with your comment, I need to say. Yeah.
Domande? Oh, Fabio.
Do you think that the DNA methylation clocks may be different between cells? I mean, or between tissues? Cells for liver or cells from kidneys, and also if there are differences between cells within the same organ, the same tissue.
Yeah, all of it is true. So when you have cells from the same tissue, for example, blood, and you measure the ages of the different blood cells, you will see slight differences, not major, you know. So example, you take a 50-year-old man and then his memory T cells may be 53 and his naive T cells may be 48. So a few years of difference, you know. Also, organs age differently. So it turns out the cerebellum in the back of your brain ages very slowly, and also the retina ages very slowly. Conversely, blood and also bone, you know, so certain cells and organs age faster, you know. So, and there was a wonderful paper recently where people looked at mice and really addressed that. I just want you to know. So people are looking on that, you know. Mm-hmm.
Thank you very much.
Mm-hmm.
Thank you, Steve. Thanks a lot.